I fundamentally believe innovation oftentimes happens in bursts. So a monoculture not necessarily fosters bursts, but you want to have those bursts to really get to breakthrough innovation. And so I do believe, biotech can play a unique contributing element in this ecosystem, small entities, agile, where there can be energy unfolded around a certain topic, so that this biotech ecosystem can actually contribute those bursts and lead us into, in a more agile way, into completely new corners of science.
This is the Nucleate Podcast, the voice of the next generation of biotech leaders.
Hello everyone, and welcome to the Nucleate Podcast. I'm Paula, and I'll be your host today alongside Anastasia. In this episode, we're joined by Dr. Uli Stilz, head of the Bioinnovation Hub at Novo Nordisk, based in Boston. He leads strategic partnerships and early-stage innovation in cardiometabolic diseases. Dr. Stilz has a background in chemistry and molecular biology. He earned his master's at ETH Zurich and completed his PhD at the Max Planck Institute, followed by a postdoctoral fellowship at Caltech. He later moved into industry to help translate scientific discoveries into real-world therapeutics, contributing to over 60 preclinical and clinical drug candidates during his time at Sanofi. Since joining Novo Nordisk in 2014, he's continued to focus on innovation, and in 2020 he launched the Bioinnovation Hub to bring together biotech agility and pharma expertise. His work focuses on emerging technologies such as RNA editing and AI-driven drug discovery, with a strong emphasis on interdisciplinary collaboration. Dr. Stilz is dedicated to advancing metabolic disease treatments and fostering biotech industry partnerships that accelerate drug development. We will dive into Uli's journey from academia to industry, the uniqueness of the Bioinnovation Hub, and how emerging technologies are shaping the future of drug discovery. Let's get into it.
Thank you very much for joining the podcast, Uli.
Thank you for having me, Anastasia and Paula.
We are super excited and also looking forward to the conversation and learning from you. But before we dive into this, let's go to the beginnings. So could you take us back to your upbringing? Were there any early influences such as your family or your environment that shaped your curiosity for science?
That is a good question, Anastasia. And I was born in southern Germany, and I grew up in a lively family where many things were discussed. And, I think one of the early memories I have where I got excited about science was that my uncle was a physician. And from him I got as a gift, I think it was a Christmas gift, a microscope. I probably was around 10, 11, 12 years old. And then with this microscope, I started to, discover other, smaller structures which you cannot see with your eye. And over time, then also I felt like, I needed some chemicals and dyes to get an even better picture. So I came from basically starting from, early thinking about biology to then more to chemistry and the curiosity to better understand the world and how it looks like. And so that was for me the starting point of scientific curiosity. And then when I was about 14, 15, I had the firm view as a high school student that I should be studying chemistry.
So I'm curious, it's a very familiar story, I think, for a lot of people who work in sciences or went to sciences, that they, at some point, they had a microscope or they discovered something, they saw something, and they were just like, we're so fascinated by the whole world that they dove into the amazing world of sciences. But what I'm right now wondering is if there are any stories, any failed experiments during your childhood that you would love to talk about.
Of course, I started to build a chemistry lab in the house. And I think, that triggered also some family emotions when certain smells went through the house. And I remember also one time where I was working with some, quite aggressive acids which were dropping on my shoes. And afterwards, I had some holes in my shoes. So that was my first early experiments in terms of chemistry, that you actually has to be also really mindful and careful how to work. But so that are very early, memories, how I started to discover science and do first experiments, and also learned about that you have to actually be really precise.
This is a very interesting story. But thankfully, no bigger issues like explosions or setting fire to the house. So, great. So throughout your career, you switched between multiple disciplines. So first biology, then chemistry, then you did some biophysics, and ultimately you went to drug discovery that we want to talk to you later. So could you walk us through how you navigated those transitions?
Yeah, and I'm happy to speak to it, Anastasia. And I would say, certainly it wasn't organic, but it was sometimes in big jumps. And what I mean by this, of course, I started studying chemistry. I did spend 2 years in Freiburg, and then I went to ETH in Zurich. And of course, it was very different than what I first assumed as a high school student. A lot of math and physics and quantum mechanics. So also understanding the theory and the theoretical background, and then in parallel learning the experimentation. But then after 5 years, when I had my master at ETH, I felt like, at the time, chemistry was very separate from biology. So I learned a lot about physics and mathematics and chemistry and how to make molecules, but I had basically no insight in biology, beside what I was reading at the site. And so I felt, I really wanted to learn and better understand biology, because I also felt like that's important to understand this in terms of if I ever wanted to make an impact on human health. And then the best way to learn it, I felt, was to get a PhD in biology or biochemistry. And so I started to interview, and I ended up interviewing with Dieter Oesterheld at the Max Planck Institute, or the Max Planck Institute for Biochemistry in Martinsried, close to Munich. And it was a fascinating interview because Dieter Oesterheld, of course, very quickly realized in this interview that I basically knew nothing about molecular biology and cell biology. At the same time, he had a unique way to react to it, where he looked at me in the eyes and said, I always like young people who want to learn something new. And so you can join me. So I offer you a position. And then he created his own way of creating a barrier and to test me. He said, but I don't have funding for you, so you have to find a fellowship. And so I went out to get a fellowship, and I joined him in 87 in Munich, and then started to learn about molecular biology and about cell biology and protein chemistry. And at the time we worked and studied the photosynthetic reaction center. Now coming to your second question, how I came from chemistry to biology to biophysics. And this photosynthetic reaction center, we wanted to understand what is the role of the protein environment in facilitating the electron transfer from the chlorophyll molecules. And so we made mutations and structure function study in this protein. And to understand it, we needed femtosecond spectroscopy. And that, of course, was really high-tech physics. And so we collaborated with a group at the university in Munich, and I learned a lot about physics and biophysical methods. But I also, and that I think was important for my later career, I also learned how to collaborate across disciplines. So certain disciplines which I learned, and certain which I learned how to collaborate, but not being the expert, but knowing enough about it that I could talk to the experts.
That's very interesting. Could you elaborate on, you mentioned you learned how to work interdisciplinary and with different profiles, which is a very common theme, for instance, for physicians to work with computer scientists to create some AI tools, or for researchers to talk with physicians. And basically, you have the same goal in your mind, but you are trained to think differently, which is great. But oftentimes there's some pressures or like some misunderstandings coming up because you essentially don't speak the same language at some point. So can you maybe elaborate on how you learned this and any advice for our audience on how to deal with those situations?
Yeah, I think, Anastasia, for me, of course, it started actually first learning chemistry and then learning biology. So I already learned two languages. I learned the language of chemistry, which is very much structure focused. And I learned the language of biology, which is mostly focused about, understanding function. And so I started also to appreciate that different disciplines have actually different languages and the crosstalk between the two. But then I also started to learn and appreciate when working across disciplines and disciplines I'm less familiar with, that one of the most important things is actually to be curious and to ask questions, to understand where others come from. And that relates to science and scientific discipline, but it also relates even more so to humans, where different colleagues come from in terms of what's actually important for them.
And that's a very great insight, this thought of curiosity and being open and actually having a dialogue. So essentially, you did this whole career, you've switched a lot of specialties, you mastered those specialties, and you learned a lot throughout. So at what point in your career did you say, oh, I will switch to industry? Was it something that you've been thinking for a long time, or was it more a chance that you took at some point?
Anastasia, I didn't know for quite some time, right? I went from Zurich to Munich to get a PhD, and then all during this time, I wrestled with the question whether I should stay in academia or go to industry. And because I couldn't decide, I decided I first do a postdoc and went to the United States. So I went to Caltech, to California, and did spend 2 years in a research group, which was really at the interface between biology and chemistry. And so we started looking for specific molecules which could recognize DNA in a very specific way. And there was a vision we could develop a completely new class of drug molecules, or a more generalized approach to drug discovery by recognizing DNA in a specific way. It was a big picture vision in the early 90s. Much later, CRISPR-Cas9 was discovered, which then actually enabled it. But I think it was during these 2 years that I more and more felt like I want to really make an impact on human health and move into biopharmaceutical industry. And mainly because, I think the key driver was I wanted to be part of a journey to find new medicines.
Since our audience are students predominantly, or graduate students, who are struggling with the same question, what do I do now that I am about to graduate? So how did you actually go with this transition? Because pharma industry is also a big space. So how did you decide on which specific area in the pharma industry I want to go into? And how did you, like, maybe it's a little bit like a game plan for someone who's actually in this position and says, oh, I want to go to pharma. So what would you say what this person should do?
My journey at the time was to go in one of the large pharma companies and start in a research department. And I think, retrospectively, that was actually a really good starting point, because I was surrounded with a lot of experienced colleagues and deep domain expertise. And I learned so much from my colleagues in an environment where I could work in the lab for the first 4 years of my career, and then got involved in a number of programs which, luckily were successful, that we found some molecules which we then wanted to move towards the clinic. So I moved with these projects and learned about what does it actually take, what are all the important questions to think about, from what is the biology we want to intervene with, what type of molecules are we thinking about, what is then the clinical development path, and what are all the questions, what it takes to scale a molecule, to think about how to study toxicology. All those questions I learned in those 4 years, being surrounded by many colleagues who helped me, who trained me, who were patient with me, that I asked many questions and didn't know many things. But so I think training in one of those larger pharma companies for scientists over the first 5 years is, I think, a really good training ground. Now, I think, Anastasia, was also true when I started a little bit more than 30 years ago. It was a different environment than it is today. It was mostly characterized by larger pharma companies, and biotech was only emerging. When I was at Caltech in '91, working with Peter Derwin, he was sending me to one of the companies he had founded, which was a company in South San Francisco. And I arrived, it was about 30 people in half of a building. Now, many, many years later, 25 years later, I came back to this company, and it was like 15,000 people in South San Francisco. And the company we all know today is Gilead. But at the time, it was one of the first starting small biotech companies. So I think the biotech industry just started to form around this time. In the mid 80s, late 80s, early 90s. So it's much more diverse nowadays than it was 30 years ago. But I still believe to learn drug discovery, some of those larger organizations provide a really good training ground. And I would think, for young scientists today, might be a good starting place to train, and then thereafter to venture in many of those smaller biotech companies.
Yeah, that makes a lot of sense. So actually, first of all, learn how to play, and then go out and have the best game that you can have. So I would jump a little bit to what you're doing right now. You are in Novo Nordisk, but you have a very special position. So you are the head of the Bio-Innovation Hub. So first of all, for those listeners who might not be familiar with this, can you briefly describe what it is and how it is different from a typical pharma R&D unit, and what's the special sauce in it?
So the Bio-Innovation Hub is at its core an R&D unit, right? Because our focus is to develop new medicines or new experimental medicines. What is different to many other R&D units, which are focused on internal research, is that the Bio-Innovation Hub, everything what we do with partners. And we are very deliberate about it, because what we want to do is we want to have a team, or that's how we designed the Bio-Innovation Hub and the team here, to have a deep insight into cardiometabolic diseases, and then work with entrepreneurs and entrepreneurial units around us in Cambridge, in Boston, in the wider North American innovation ecosystem, to develop jointly ideas for next generation therapeutics, where we bring some expertise to the table, and even more importantly, our partners bring unique skills and capabilities. Either they bring a very novel idea which needs validation and then acceleration, or they bring a platform capability which so far has not been applied to solve and address a question which could be relevant in the cardiometabolic space, so that we help pivoting this technology into the cardiometabolic space. So that's how we have focused on building a unit which everything what we do is focused on co-creation. How can a partner add something and bring something which we don't have, and we add something the partner doesn't have, and together we then can go into wide space of innovation which has not been conquered yet. So that's our focus and makes us unique. And in some way, as we conceptualized it 6 years ago, we felt like, in Novo Nordisk, we had an innovation ecosystem, very strong internal research, and that has been the historic focus. And then over time, we started also more to emphasize business development. So how do we find what others have invented independent of us, and then can decide do we want to actually bring it into our pipeline, and a very capable corporate development group doing this. And then we felt the Bio-Innovation Hub is the third pillar of innovation, where we focus on shaping the market and driving more innovation in the cardiometabolic space, and doing this by creating a team here in Cambridge, which then can work with this ecosystem to be really active shapers of the environment so that more ideas flows into the cardiometabolic space, because we felt there is a huge unmet medical need. So many people being impacted by diabetes, obesity, cardiovascular disease, and still many open questions where we felt more innovation will be needed to help those people.
Yeah, that's very exciting. Also, like seeing this unmet medical need and actually setting something up that helps to cover it and helps to address it. So you mentioned you work a lot with partners and you have a lot of very, very high-ranking academic partners. How do you decide who you partner with? And maybe a little bit on what's the best partner? How do you decide who is the best for you? And how do you go about it?
I think, Anastasia, that's a multifaceted question. So there is one component to it. What are the big questions we want to address? Mostly we have focused on three, and I can speak to it in a minute. So one question in diabetes, as an example, a chronic disease, a progressive disease. We all know now, it's a little bit more than 100 years ago that insulin was discovered in Toronto, which was and is a life-saving medicine. And then a few more drug classes have been discovered to treat type 2 diabetes. What is common to them that they primarily are symptomatic treatments, lowering glucose, and then also some of them providing additional benefits. None of those drug classes yet really, get to the root cause. So diabetes as a disease keep progressing, even on those treatments. So what I mean by this, once a physician endocrinologist has a patient coming to his practice who then is diagnosed as a diabetic, there's no way back. Once you're diabetic, so the disease keeps progressing, but you cannot go back. So one of the big questions we are asking is, can we get to the root cause of the disease? Can we understand the biology and the root cause? And what could enable this? And by understanding this and hopefully, diving closer to the root cause, could we then find disease-modifying therapies? So that's one question. And then on these questions, then to think about, what could be new paradigm? What could be envisioned as there could be new paradigms? Was driving us at this specific question to certain partners. So, for instance, we said, we want to work with the broad on this question, with researchers at the broad, and also with researchers across the river at MGB or Mass General Brigham, so that we have a combination of basic science and clinical science, of human genetic data, of understanding the disease from the human biology side, but then also having the tools at the broad to deeply study the biology and combine with this data science. So can we segment patients and in that sense sub-segments get to the root cause of what is driving type 2 diabetes? So the partner selection here, to answer your question about how we felt there is unique scientific excellence at the broad, at MGB, and in this ecosystem where we felt like that we want to tap into and then add our expertise to it. Then a second question, being focused on is this question. There is insight into biology we believe is really important in diabetes or obesity or cardiovascular disease, where we believe we have a fairly good understanding of the biology, but we don't know how to design a molecule to modulate the system. And certain things we can do with small molecules, but other things we can't. Certain things we can do with biologics, antibodies, and peptides, but we have not yet solved the question how to get them into the cell. So not all questions can be solved with the modalities which are, today, day-to-day, well-established modalities. So we are on the constant outlook here in this ecosystem for new emerging modalities. And could those modalities unlock biology? We strongly have a strong conviction is important, but we haven't found a way, how to address it. And so that, as an example, has led us last year to start a partnership with a company called Coro. And Coro, I was very intrigued because core had started to build a platform around this question: Can you actually, capitalize on a system which was found in lower species, and with indication it also exists in humans, that the messenger RNA is not constant but can be edited. So that actually is the paradigm which we all learned in molecular biology. We transcribe DNA in messenger RNA and transcribes into protein sequence. That the DNA sequence actually translates one-to-one into the protein sequence is actually not really true. But there's a step in between, and the possibility to change the messenger RNA sequence, and in this way also to change the protein sequence. And that allows us to modulate proteins by modulating messenger RNA sequence. And that we found very fascinating, because that is basically then a pharmacological tool using small oligonucleotides, quite similar to siRNA. siRNA using the Diaz complex in RNA editing. It's a different system, an ADA system, to deaminate adenosine into inosine, and then it gets translated into a different base pair. So it allows single point mutation in the messenger RNA and then to express different proteins. So we felt like, okay, that is really fascinating, and can, as we started to think more about it, lead to many unique therapeutic concepts which could be developed to modulate proteins and their properties, to repair point mutations, to intervene with how transcription factors work. So we found that's actually a quite attractive platform, and so we started a collaboration on a very specific project where we felt this is important biology, and we felt there's no other way to get to it, and messenger RNA editing could be a way to address it. So very innovative, novel platform. For me, of course, in a way also nice to see, since we started this collaboration, that there were some first human proof points that actually the system can also work in humans, which makes me even more excited, right, about this platform. So that's the second one, and we keep looking, right, for platforms which could unlock new biology. And then the third one, I think also important question for us, is in this chronic disease space where so many people are impacted, that it's vitally important that actually also to look out for emerging platforms which would allow to get to infrequently dosed therapeutics, which could improve compliance, right? So people living with a certain condition don't have to think about every day to take a tablet, but maybe it's only kind of 2 shots a year, or very infrequent dosing, so that could improve compliance and treatment outcomes. It could improve sustainability, but it could also make an important contribution to scalability. I certainly realized over time that it's very different to think about a human condition where 100,000 people are impacted, to a condition where 100 million or 500 million or close to 1 billion are impacted. Now in diabetes, it's today more than 500 million people who live with diabetes. Obesity, it's over 800 million today, and the numbers are growing. So whenever we think about developing a new medicine, that we can actually also scale it and make it available.
Yeah, that's a very very broad overview, and also very good to know and to hear. So you mentioned you're always on the lookout for new modalities, for something that is not there yet to address, as you mentioned, this unmet need, especially of those diseases that right now they have treatment. Some patients they do well with these treatments, but essentially it's not always like that, and there's still this unmet need to actually make it better and maybe address the root cause. So I would think that you see a lot of innovation, a lot of new methods that are coming out of academia on a very frequent basis. How do you decide which ones are the best ones, the most promising ones? And what's your decision process there? Because, kind of just take everything that you see and that you find cool is not the best approach on getting to the goal.
No, and I think, Anastasia, this is maybe one of the most difficult questions, right? Because there are so many areas and ideas of emerging science, how to scan, how to select, how to think it through, how to actually think it through what could be the implication oftentimes that's not really visible when an early piece of science emerges, what could be then a real life use case for it. So I think, we look at many things and then we discuss it with the scientists involved, either in academia or in young biotechs. And then I think also important for me is that the unit, like my unit, is also very experimental. So that we are not, trying to assume we know everything, but that we actually engage with emerging science once we believe this could be important and generate data. And so we created also different type of partnership framework. So for instance, we created a partnership framework which we later called an co-creation greenhouse. And we called it co-creation greenhouse because we felt like, oftentimes, there's an early piece of science and it's not so clear what it can lead to. And then large corporations like us wait and say to those investigators, come back in a year from now with more data. But oftentimes those investigators don't have the means to actually bring it in a direction where it could address a real life use case. So we said we actually have to engage earlier and create then a partnership framework where we provide some guidance. We typically spend a week or two with young investigators then to think about, basically doing a Google design sprint, what could be a real use case, and then develop a joint research plan over 3 to 6 months to get to some data points. And then we fund it to a certain level. And importantly, we actually don't make a contract. We agreed we want to actually be quick in the lab. We don't want to have a long negotiation about a contract, but we say, we fund it and then generate data. And then after, we of course work from the assumption that once we have worked together 3-6 months, that we also, stick together if we are successful. But so that has led to cases where within, between the first that we met to first experiments in the lab, it was not more than 2 weeks.
That's unheard of, especially when you think about academia and how slow all the processes are, and especially if you go into some kind of legal negotiations, it can take weeks, months, years. So it's very refreshing to hear that that you have this trust, I would say, in your partners, and that you operate under the assumptions that you have this goal in your mind and you want to reach it as fast as possible without engaging in who gets what or kind of some kind of small things that do not make it faster for you to have some certain questions answered. So my last question would be, so we've talked a lot about innovation, about addressing certain disease, certain unmet needs, and you've seen certainly a lot. So what's the most exciting thing, topic, new modality, whatsoever that you what excites you the most right now?
It's along the 3 lines what I discussed. What excites me most, seeing some data that actually, we can start to sub-segment chronic diseases like diabetes. So there I see some, really first emerging exciting data. And that gives me hope that we can get closer to the root cause of diabetes as a disease. So that I think is really, really exciting. I see, some programs where we engage in new modalities. I spoke to Core. We also made a partnership with a company called Metaphor, where we see this crosstalk between, AI, so computational methods to design molecules, but not so much on a structural base, but on a functional readout base. Can we understand GPCR signal and how then to design molecules and antibodies in a very specific way where we believe we can develop next generation obesity treatments? That I think is really fascinating from the crosstalk between biology, functional readouts, and then AI methods to design molecules on a functional level. And then I see some programs where we believe, they could be highly scalable and truly disruptive. Very early today, right? It's very experimental, but could, for instance, and that's, I think, a very emerging field we are looking into right now, is there potential actually that the body could make some of those therapeutics?
I wanted to build a little bit on that idea. We're talking about just basically this entire spectrum of different platforms that can potentially address cardiometabolic diseases. And something that is, has gained so much traction is all of this wave of precision medicine and personalized therapies. We know, cardiometabolic diseases are very complex and very multifactorial. So how do you see precision medicine sort of like shaping the future of these treatments? Can we truly move beyond that one size fits all and really get very, very personalized in these types of therapeutic areas?
I think, Paula, that's a very good question. We saw, of course, in 2000 when we had completed sequencing the human genome, significant progress in some therapy areas, mostly rare disease and oncology. Therapy areas which primarily are driven by genetic mutations. And that, of course, we could find by sequencing the genome. In the chronic disease space, I think it's more complex. But I do think we start to see, and I talked about diabetes and what we do with the broad, that we start to see subsegments, subpopulations, so that, at least we can get to a place where we say, diabetes is probably not just one disease, but there are subtypes below the broader umbrella of diabetes. And understanding those sub-umbrellas could lead us also to understand more specifically what is driving the disease in these subpopulations. So that I can see happening over the next years.
So identifying sort of like this segments within these very complex diseases.
Yeah.
Well, maybe a follow-up. How do you feel like, for example, there's some like gene therapies like on the pipeline also for diseases as diabetes. How do you think we can like avoid the same challenges that when you get into like this huge market disease, like it's different to treat a very rare disease, a subset with a gene therapy versus, trying to deploy a solution like that in such a prevalent disease like diabetes and, like the cardiovascular market?
Yeah, I think, Paula, gene therapy, of course, I can clearly see in rare diseases where it's specifically known what the mutation is. When it comes to a chronic disease, that's more complex, right? Because we cannot pin it down to one gene mutation. And then the other question is, of course, every new modality needs to prove itself first in real life. So I felt like, and that's why I made this comment about Coro, which I found quite fascinating, because there we don't modify the genome. We edit the messenger RNA, so it becomes a pharmacological tool. And my personal scientific conviction, and I might be right, I might be wrong, is that a pharmacological approach at this point in time, as much as we know, puts us maybe in a little bit more of a sweet spot to enter into the chronic disease space, and if we start modifying it at the DNA level.
Well, I think with that, we have definitely covered a lot now until kind of like drug development and what we can be looking forward. How about we switch gears towards the actual like innovation systems? How pharma and just R&D in general works. We cover a little bit about how BIH is sort of disruptive in a way. So here's a question. So if you could redesign the biotech and pharma R&D model from scratch, what would you change?
Oh, that is a big question, Paula. But maybe I can just add a few thoughts, right, to this. So, I said earlier during, our conversation today is that I felt like it was really good to join a large pharma company because there's a lot of expertise and I could, as a young scientist, learn a lot. And I continue to believe that it's true that these large pharma companies can have a unique place in this innovation ecosystem to really be deeply knowledgeable about therapy areas. What is this disease biology? What is the current standard of care? What is the unmet medical need? How would we and think about clinical trials? What improvements are really meaningful for people living with those conditions? At the same time, these large organizations also have a tendency then to become quite homogeneous. And what I mean by this is to have one approach to innovation and then one process how to progress and think about, we have one pipeline, we have 100 projects or 50 projects and we have one pipeline and we execute on them all in a similar fashion. And I fundamentally believe there is a benefit in this, but not necessarily always the best approach to innovation, because I fundamentally believe innovation oftentimes happens in bursts. So a monoculture not necessarily fosters bursts, but you want to have those bursts to really get to breakthrough innovation. And so I do believe, say, biotech can play a unique contributing element in this ecosystem, small entities, agile, where there can be energy unfolded around a certain topic, so that this biotech ecosystem can actually contribute those bursts and lead us into, in a more agile way, into completely new corners of science. And then to think about in this ecosystem how to get these different pieces actually into not separate entities, but how to connect pieces of this innovation ecosystem to gain more product or to gain additional productivity, and actually helps that new ideas can actually very fast get to clinical trials and finally help people living with those conditions. So maybe those are some of the questions, right? Or how to create more bursts in the overall ecosystem, and then how to help in the ecosystems that energy get be funneled on, that really exciting ideas get also the support and the intensity to quickly go through clinical trials.
So smaller biotechs are like working towards, smaller biotechs and big pharma companies are already like sort of like working towards this goal through different approaches. And that way there's like this entire diversity that can move things forward faster.
Yeah, and I think, in small biotechs there is real intensity, right? Because the whole organization is focused on one topic. And so there is agility and also dedication then, because if you work on one topic, you have to succeed on this topic, right? Because that's what your organization is all about. And I think, Paula, it's for me fascinating to see these large pharma companies are organized in therapy areas and drive projects in therapy areas. But they're not really set up, from what I've seen in my career, they're not really so well set up to develop new capabilities, for instance, new modalities. And of course, this industry came out of the chemical industry, the dye industry, and was starting with small molecules. That was the birthplace of the pharma industry. And then this industry stick to small molecules until monoclonal antibodies were discovered. But the breakthrough did not happen for the most part in large pharma companies, but in small biotechs like Genentech and alike, Amgen. And the same repeated when it came to antisense and later siRNA. The breakthrough at the end, even though the big companies all tried, or many of them tried, as a breakthrough happened in Alnylam, in Ionis, because all the energy was focused on creating this breakthrough, because that's what those organizations were focused on. And so then it creates an ecosystem where some of those companies build new capabilities, and they benefit also then from partnering with the large pharma companies. It's noticeable many of the biotech companies which at the end were, or most of them which were at the end successful, had the insight strategically to early partner.
That's definitely very insightful. And, as all of this, I guess this is something inherent to science. Progress is non-linear. You can devote a lot of time and effort to this one thing, and then it can fail. And there's setbacks on the road. So I would love if you can share a story with us, or experience of a case where maybe it can be back from a research project or maybe a partnership that you thought it was going to go better, and then maybe there was an unexpected challenge. And how does the team adapts around that? Basically, like, I want a message to our listeners about how do you adapt from those challenges and from maybe failure?
Yeah, and I think, Paula, that is a really good question. And I'm thinking I give you one example, not so much a specific science project, but more a partnership project, and how actually to create energy between organizations. So we started about 5-6 years ago. We started a dialogue with Flagship, and the feeling was, a partnership between Flagship and Novo Nordisk could be really impactful, because it's two very different organizations. Flagship very focused on creating platform companies like Moderna, which is very well known now since the pandemic. And Novo Nordisk having this strong and very intense focus on diabetes and obesity. So therapy area focus. So the notion was at this intersection to, basically orthogonal innovation models. One focused on platforms, therapy area agnostic, and Novo Nordisk focused on obesity and diabetes. And so at this intersection that we could actually productively create new insights, innovation, new product concepts. So we met and we discussed, and then we said, but we don't want to work just with one company in Flagship's ecosystem, but can we make something? Can we make an ecosystem partnership? And that was very appealing. And, we had a clear vision in our mind. And then we started thinking it through, and from a vision point of view, how would we translate it into a business framework? And we tried to solve everything in one go. And then we started to stumble over very specific question. How would we actually go about intellectual property rights, as an example? How would we actually define this in the business framework when we don't even know what the product concept is, and then we don't even know what the company is? So we tried to solve it in one big framework, and then realized that's actually not really working, right? So then we pivoted away from it and said, no, this has to be much more adaptable. And so we kind of pivoted away from the first idea how we can do it to get to a much more adaptable framework. So we really inserted creativity in creating a business framework. So we realized, it's not only needed to be creative on the science side, but it's also important to be creative on the business side and get those two things talk to each other. And now, we created a really impactful partnership where we have created a number of important product concepts which we are driving now. But there was some iteration and learning, and now I think the spirit between the two organizations is actually continuous learning. Right? How do we work together in the best possible way to create significant impact for people living with obesity or diabetes?
Yeah, so definitely there was common ground. You both wanted to collaborate, but then being open to iterate and make changes, and not like this very rigid.
I think what's important and what I keep emphasizing is actually to first agree on a vision. How does good look like? And that becomes a North Star, which then can bring two organizations together and to actually go through the struggle, right, to find out. Because you could also say, it doesn't work, right? Then we walk away. But to have a clue in terms of a joint vision, which then provides enough energy and motivation to say we actually want to solve it.
I love that story. I think definitely we can learn a lot from it, and honestly apply it to so many different parts of our lives. I think not only to partnerships, but also the way that you deal with like challenges on a day-to-day basis and trying to keep that North Star. I love that concept. So Uli, tell us a little bit about, what's next for BIH. We talk a little bit about what excites you. We talk about like these subsets of patients that you think maybe areas where we're gonna see some breakthroughs, but yeah, just tell us a little bit more.
So I think, most important right now for me is, we have now quite a number of really important programs where we have a deep conviction. So to drive those towards the clinic, right, that's a key priority to really capture value for many things we have, ideated and started. And then there is constant flow of, science is moving so fast of new ideas. I'm very fascinated. I'll give you a few topics. I'm very fascinated by, the impact AI can have on drug discovery, both to design molecules, which in my mind may be more nearer term, but also to understand biology, which might be even more complex, simply because we humans are complex. And to understand this, but AI, I believe, will have a significant impact. So how can we use actually AI and machine learning tools to understand biology and then become even more, deliberate in terms of where we intervene in a disease process? So I think that's I have a deep conviction that this is an important journey to invest and be very deliberate about. Then I also believe this broad topic of genetic medicines is really important to make precision medicines, both to downregulate and to upregulate genes and gene clusters to find new treatment options. And then I think this crosstalk between also, human genetics, human-based biology, and AI is a really important area. So understanding human biology as a key focal point and driving chronic disease to the root cause, so that we can find disease-modifying therapy. So that are a few themes. And then if it comes to specific question, oftentimes I feel like, every day, I'm involved in exciting discussions, and I'm probably most excited about the things I don't know today.
Yeah, that's a great point. So, okay, yeah, definitely a lot to look forward to. And now that we talk a little bit about the future, maybe we can, go to the past in the sense of like when you look back in your career, and considering our audience, what advice would you give to young scientists, entrepreneurs, even policymakers who really want to make an impact, which seems like this has been like the core value of your career, trying to make an impact on patients. So what advice would you give to them that want to drive this real change in healthcare innovation?
Yeah, Paula, to young scientists, I would say, important to be curious, to keep learning and adding to what you know. I made many jumps in my life, right? I jumped from chemistry to biology. I jumped from the science side to the business side, how to bring entities together. I had a number of different roles, which took me completely out of my comfort zone. And so I would say, not to be afraid to make a jump, to actually experiment with yourself and accelerate your learning journey. So that's maybe one of the key recommendations I would have for young scientists when they think about their own scientific career. For the larger ecosystem, I think, that you continue the journey to build unique capabilities in different organizations and then find a way, how we can bring this together in a collaborative environment. How can we jointly drive value? And then oftentimes in business negotiations, sometimes I say this with a smile, but to be also insightful, that it's when we negotiate, not so important to always win, but to think about the other side. What's important for the other side, and then find actually, I would sometimes call it when the negotiations, there's 100% in the negotiation to be distributed. It doesn't really matter if you get 49 or 51%. It's much better to negotiate in an ecosystem. How can we make out of 100%, 110? And then it doesn't matter so much if you have 54, 56. And I think that can only happen with empathy and understanding, asking not so much the question, what's good for me, but asking more the question, actually, what's important for the other side.
I think that's definitely like something that resonates. And it reminds me of like this one about like, it's not you against me, it's us against the problem. And keeping in mind what's really important and why on the first place two parties are having the conversation. So
Yeah.
With that, maybe like a final question. If there's something, if there's a message or an insight that you hope that the people take from your career, which, I mean, it may overlap a little bit what we just covered, but like maybe something that you want people to think about when look at your story.
I think, Paula, it's very much what I said before, to feel comfortable to take risks and to take some jumps into the unknown, and actually to feel comfortable in the space of uncertainty. Because in my mind, the space of uncertainty is actually where the opportunities are. That's where something new can be discovered. If we are certain about something, then we know already how to do it. But to feel comfortable, to be in a space and holds attention, uncertainties, and to start to pioneer.
Well, thanks so much, Uli. I think this has been really, really, really insightful. I know our listeners are gonna get a lot from it. And yeah, any final thoughts?
Just thank you, Paula and Anastasia, for a wonderful dialogue today. It was enjoyable talking to you.
Thanks for listening to the Nucleate Podcast. Be sure to subscribe if you'd like to keep up with future episodes. See you next time.